Hi, my name is Ashley Reef. For this presentation, I will be talking to you, the general public, about a topic I personally find very interesting, newborn genomic sequencing screenings. So first, I would like to briefly go over what exactly newborn screening is. So newborn screening is a health service provided to the public in every state, and it aims to screen newborns for various serious health conditions. The goal of this is to help healthcare professionals start treatment before some of the harmful effects happen. Currently, newborn screening tests start with a hearing test, pulse oximetry, and a blood test. The blood test is usually done, as demonstrated in the photo there, with a heel stick. A sample of blood is obtained, placed onto a card, and sent to a lab. Currently, the state of Virginia screens for 33 medical conditions, such as the classic phenylketonuria PKU disorder. And a great resource to visit for newborn screening FAQs is this babiesfirsttest.org, which was established under the legislation of the Newborn Screening Saves Lives Act of 2007. So a little history of newborn screening. So that gentleman that was on the previous slide can be considered a founder of newborn screening blood tests. It was first introduced in the 1960s by Dr. Robert Guthrie to specifically identify the amino acid disorder PKU. Fortunately, PKU is easily managed with dietary changes, but if it's not identified early and treated, it can result in severe mental retardation. So in 2002, the Federal Health Resources and Services Administration's Maternal and Child Health Bureau consulted with the American College of Medical Genetics to develop guidelines for newborn screening. So at the time, some states were screening for only 4 conditions, while some others were screening for 50. The ACMG looked at a total of 81 conditions, but they determined only about 29 of them fit into a core screening panel, thus making up the original recommended universal screening panel. So by 2003, most US states only screened for 6 disorders, but since then, the importance of newborn screening has increased significantly. Today, all states screen for at least 29 conditions. And in order for a new condition to make it onto the RSUP, they're required to meet 3 minimum criteria: that the condition can be detected 24 to 48 hours after birth, when it cannot usually be detected by a doctor's exam. It has to have a test that is specific and sensitive for the condition, and its early detection, timely intervention, and effective treatment has to offer proven benefit. So a little jump into genetics and genomics. I will be giving you a crash course on this topic. So a genome has all the genetic material of a living thing, known as the deoxyribonucleic acid, or DNA. DNA groups together to form genes, which provide instructions for building the proteins that make up your body. The proteins contain all the information that makes us who we are and how we function and what we look like. This is the structure of a DNA with its nucleotides. So the genome is considered the blueprint of our body, and a human genome roughly makes up about 3 billion letters of these nucleotides, which are A, T, C's, and G's. So in 1990, the Human Genome Project launched with a goal of sequencing all 3 billion letters of the human genome in 15 years. They actually completed it in less time. By 2003, the Human Genome Project successfully completed and confirmed that we have approximately 20,000 to 25,000 genes. So let's correlate this information with newborn screening. So a little history. With the advancement of the Human Genome Project and next generation sequencing platforms, there's been a drastic drop in the costs for this. So This has opened many doors for different genomic topics, such as newborn screening. Currently, though, utilizing genomic sequencing for newborn screening is still under study. So, in some states, sequencing of a specific gene is done when a baby's initial newborn screening results show something abnormal. The DNA sequencing in these cases is only looking at one or two genes related to certain conditions and not a newborn's whole genome. So the idea of newborn genomic sequencing would not replace the standard newborn screening. It would just allow doctors to identify health conditions very early in life. Due to the growing idea of utilizing genomic sequencing for newborn screening, the National Human Genome Research Institute, or NIGRI, developed the Newborn Sequencing in Genomic Medicine and Public Health, or INSIGHT, program to help explore the implications, challenges, and the opportunities associated with the possible use of genomic sequence information in the newborn period. Some benefits and concerns with using the next generation sequencing for newborn screening. Some benefits to it could be to provide more health information than the current panel of tests, and it could potentially be used to guide an individual's lifetime of medical care, providing information on both treatable childhood diseases and conditions that occur in adulthood. Some concerns that do arise with the talk of routine newborn genome sequencing are like, what if the genome sequence reveals information about untreatable diseases or even conditions that will not occur until adulthood? Or what is the risk to the newborns that the identification of specific genetic variants, such as being predisposed to a certain disease, could lead to future discrimination? Some ethical concerns to using newborn genomic sequencing can be that some genetic changes will have implications for the health of not only the infant, but of the parents and other family members. So they might learn something about themselves in the process that they didn't know they could potentially be carrying or could potentially have later in life themselves. The interpretation of genomic data is constantly evolving, and right now it's unclear whether some changes in the genome are relevant to a person's health or not. And then while some genetic changes have immediate significance for an infant's health, other changes only influence the risk of developing the health problems later in life. Obviously, infants are unable to provide informed consent, which is generally required when testing for adult onset diseases. And newborn genomic screening raises issues of privacy and potential genetic discrimination if the genomic data becomes part of a baby's medical record. And again, a common concern regarding the new generation sequencing is consent. So typically when a newborn screening identifies a condition that directly benefits the baby by decreasing morbidity and mortality, there is a general agreement that such testing may be done without parental consent. However, with whole genome sequencing or whole exome sequencing newborn screening, it would undoubtedly require the parental consent, and this could greatly decrease participation, which would threaten the universal newborn screening. So the future of genomics and newborn screening, the whole genome sequencing and whole exome sequencing still requires some more fine-tuning. Currently, there are still many genetic variations and conditions that do not meet the standards set forth by that ACMG. The ability to screen for the additional conditions already has led some states to screen for targets that don't meet the traditional screening criteria, which has prompted more evidence-based decision-making. With the whole genome sequencing, there's the potential to provide information that does not directly benefit the newborns, such as their likelihood to develop adult onset conditions or untreatable diseases, their carrier status, and their harboring hundreds of variants of unknown significance. Currently, newborn screening takes place at a centralized state lab, and DNA testing is not a routine part of that process. So implementing the next generation sequencing testing would require a significant overhaul of equipment and personnel, as well as further educating healthcare professionals. One study I found revealed that parents had variable but overall not a very enthusiastic attitude about the potential future expansion of next-generation sequencing to include the genomic testing, thus suggesting that parental choice will be an important consideration in future screening programs. And another survey suggested that parents did have a high interest in hypothetical new generation screening, newborn screening of their newborn. So in conclusion, I feel that newborn screening in the age of genomics is a topic that will surely come into fruition in the very near future. The questions lie in whether it will replace traditional newborn screening or serve as a supplement for screening disorders within the existing framework. The RUSP should limit targets to genes and variants that have robust disease-gene relationships for which there is an agreement that the screening would be beneficial. And then consequences of the diseases screened should be clear, have well understood inheritance patterns, penetrance, and variability. The best interests of the children should remain the guiding principle in newborn screening and the basis for decisions regarding implementation of genomic newborn screening. And that concludes my presentation on the topic. And here are my references. Thank you.